In this video, I'll go through the structure and function of the liver. The liver is multi-lobed, but I'm just going to draw one lobe. First, let's look at the circulation of the liver. The hepatic artery enters the liver and through arterioles separates to feed all of the tissues of the liver. Those then join into the hepatic vein. One critically important thing about the liver, though, is that there's a second source of blood from the hepatic portal vessel or portal vein, which brings blood from the intestine, rich in material digested and absorbed from the intestine, but also bacteria and viruses that have gotten through the many breaks that occur in the intestine, and also toxins that may have been absorbed from the food. The portal vessel develops its capillaries, and those join the capillaries to form this odd structure in which three vessels come together. This is what we're going to focus on as the functional unit of a liver, in which blood is coming in through an arteriole, through a portal vessel, and leaving through the hepatic vein. Here I'm showing you an image from Wikipedia, a schematic with the liver here, and if we took a cross section, if we cut through the liver here with a knife, what we'd see is this hexagonal structure, each of which is called a lobule, and each of which has a central vein. That vein or venule will go to the hepatic vein. At each boundary is a hepatic portal vessel and an arteriole. Focusing in on one of these lobes, blood is coming in through two sources, a portal venule and a portal arteriole. Blood coming in through the portal arteriole in red moves through what in other tissues would be capillaries, but here are called sinusoids, to a hepatic venule and a hepatic vein and back to the heart. In addition, blood comes from the intestine into the portal venules. That blood joins the arterial blood. So the combined portal venule blood and arterial blood join to go back into the hepatic vein in the heart. All the blood is moving from the margins in to the center. The bile ducts are also at these corners of these roughly hexagonal lobules. Bile goes in the opposite direction. The bile has countercurrent flow. I want to zoom in on a part that just includes this. One portal venule in blue, one portal arteriole in red, one bile duct in green, with countercurrent flow, portal venule blood in one direction and arterial blood in one direction, the bile in the other, and then in the center will be the venule of the hepatic vein going back to the heart. I'll freeze on this image for a bit because you may be interested in seeing the text. The image is from Wikipedia. What we're looking at would be from one of those lobules with the central vein here and a portal venule, arteriole, and bile duct here with the blood going in this direction and the bile going in the opposite direction. This is the central vein. And so blood will be coming from the arteriole and the venule from the intestine, going to the central vein and back to the heart. Bile will be going in opposite direction to the bile duct and eventually to the gallbladder to feed into the intestine. So we're looking at this piece. So I'll sketch this as we go and talk about what we're seeing. Blood coming in through the portal venule. That blood would be going to the central vein. There'd also be an arteriole, hepatic arteriole coming in and joining, arterial blood coming in here, blood from the intestine coming in through the portal venule, joining to reach the central vein and back to the heart. The bile starts and moves in countercurrent direction, eventually collecting in the gallbladder. Now we'll do a little bit more detailed sketch to talk more about the structure. The capillary endothelial cells are, as always, very thin, but in the liver they are unusual in how big the gaps are between them. In a typical cell, the gaps may be just a few nanometers. These gaps, or fenestra, are around 100 nanometers. Remember, a typical protein is about 10 nanometers. A typical virus might be 50 to 100 nanometers. So proteins and viruses can actually move in and out of these spaces. These spaces are actually called sinusoids, in reference to the fact they're more like a sinus, something with lots of gaps and pores, than they are a typical interior of a capillary. The hepatocytes, major cells of the liver, are separated by a gap, the space of Dis, from the capillary endothelial cells. On both sides here we'd have a layer of hepatocytes. The hepatocytes secrete many of your blood proteins. These are blood proteins that may be involved in anything from clotting reactions to maintaining osmolarity or helping to buffer or transport substances. Another very important category of cells are macrophages, called Kupffer cells, part of your immune system. And these macrophages may partly block the spaces. They can move in and out. Some of them move and others are stationary. Their function is to capture materials coming in through the portal vessels, which might be bacteria, might be viruses, and those will bind to a Kupffer cell and be phagocytized, taken into the cell, and destroyed. Also coming from the intestine are immune complexes that may be antibodies bound to toxins in complexes. These would be smaller than the viruses that I've drawn here. They also will attach and be ingested and destroyed. Kupffer cells are a major part of your immune system. This makes the liver a major immune organ. In addition, because so much blood is moving through from the liver, they also encounter other immune system cells, lymphocytes. Kupffer cells and other immune cells also produce signaling molecules of the immune system, so immune system messenger molecules. I mentioned these hepatocytes, but we haven't talked about the gall yet. Hepatocytes line the ducts that feed into the bile ductal, and these send the bile to the gallbladder and then eventually to the intestine to aid in breaking fat droplets into much smaller drops. The hepatocytes on the opposite side from the space of Dis secrete bile, especially salts of cholesterol. Remember, cholesterol is these four rings, and they'll be charged at one end and nonpolar at the other. Bile is being produced and transported in. This is a countercurrent exchange system. The bile is picking up some metabolic waste products or toxins to be sent back to the gut. Another extremely important function is to recycle the bile. The bile is secreted into the intestine to break up fats. Most of that bile, 90% of the bile, is recovered in the lower part of the intestine and comes back in the portal vessel. And I'll use B in the sketches to refer to bile salts or salts of cholesterol. So the bile is moving in opposite directions, and this is a typical countercurrent exchange. Consider bile salts, salts of cholesterol, coming back from the intestine at high concentrations and transfer to the bile ductal. So as the bile salts move in countercurrent through the sinusoid, The bile moving in the opposite direction will contain a high amount of bile salts already picked up here. It's encountering fluid going in the opposite direction that has even more bile, and so some of that bile gets transferred, adding to the bile in the bile ductal. Some of that bile got transferred. At some other area we have a bit less. I'll draw 5 Bs here. It's encountering bile that again has a little less bile, so some of the bile salts get transferred. And again, that encounters an area that has even fewer bile salts, so some bile salts are transferred. Therefore, bile salts entering the hepatic vein are at a much lower concentration, perhaps 10% or less of what it was coming into the hepatic portal vessel. Most of the bile has been sent back to the intestine. On each cycle, about 10% of the bile is lost in the feces, 90% is recycled, and the remaining bile salts enter the hepatic vein to the heart. But eventually those bile salts arrive back at the liver in the hepatic arterioles, re-entering the hepatic sinusoids. The blood moving toward the central venule always is encountering on the opposite side of hepatic portal vessels bile that has a lower concentration of bile salts than the blood. Therefore, there's efficient passive transfer of bile salts, salts of cholesterol, into the bile ductal and then the ducts to the gallbladder. And at each stage, the countercurrent exchange makes for a very efficient transfer. This image shows a little more information. It's from a special feature on liver immunobiology, and I have some text here that I've taken and edited out of that article. It talks some about regeneration of the liver and its amazing capacity to regenerate if it's not been too badly damaged. Here are the structures I talked about: portal venule, arteriole, the blood coming together, the Kupffer cells that are part of the immune system, the space of Dis, the endothelial cells here, the bile moving in the opposite direction, and the central vein. This is a little bit more advanced text about the fenestra, the perforations or gaps, and the interaction of Kupffer cells with some of the other cells involved. And finally, a little bit more, this I've taken directly. What I wanted to note is collagen that's supporting the capillary endothelial cells, stellate cells that are producing some of these proteins, Kupffer cells on the interior. Here's another white blood cell, a natural killer cell.